Inheritance of traits in Mendel's pea plants was very simple. However, inheritance of many traits in other plants as well as animals is much more complicated. Here we discuss many of these exceptions to Mendelian genetics. If we want to study transmission of some of these human traits, we need to use a pedigree, which traces historical and medical records, since direct crosses of humans is not possible as they were in pea plants. The traits that Mendel studied had only two possible alleles that showed two distinct forms, either the dominant or recessive trait. Examples included yellow or green seeds or round or wrinkled seeds. However, we now know that the inheritance of many genes in plants and animals, including humans, is much more complicated. One of the exceptions to complete dominance in Mendel's pea plants is incomplete dominance. In this case, the heterozygous phenotype is intermediate between that of the homozygous phenotypes. In Mendel's pea plants, a heterozygous plant displayed the dominant phenotype. An example of incomplete dominance is flower color in snapdragon plants. If you take a look at the scenario below, a plant that has two alleles for red flowers will have red flowers. A plant that has two alleles for white flowers will have white flowers. However, a plant that has an allele for red flowers along with an allele for white flowers will have pink flowers, which is the intermediate phenotype. This means that red is not dominant to white and white is not dominant to red. Take a look at how the genotypes are written. Despite the fact that the letter R is used, this does not mean that red is dominant. There's no lowercase letter to indicate a recessive allele. Instead, an R without an apostrophe refers to the red allele, while an R with an apostrophe refers to the white allele. When a homozygous red flower snapdragon plant is crossed to a homozygous white flower plant, all of the offspring are heterozygous with pink flowers. If the heterozygotes are crossed, there are three phenotypes present in the offspring in a 1:2:1 ratio. One of the offspring has red flowers, two of the offspring have pink flowers, and one of the offspring has white flowers. While an individual carries only two alleles for each gene, more than two alleles for a gene may exist in a population. This is referred to as multiple alleles. There are many examples of this in humans. For example, think about the eye color gene. Different alleles include brown, blue, gray, hazel, or green. Another example is the ABO blood group that has three alleles that yield four different blood types. Once again, this is different from what Mendel saw in his pea plants. In Mendel's pea plants, there were only two alleles total for each gene: violet or white flowers, green or yellow seeds, round or wrinkled seeds, etc. Another exception to Mendel's rules is codominance. In this case, heterozygotes display phenotypes of both of the homozygotes. An example of codominance is in blood type. But let's start at the beginning. Red blood cells have glycoproteins on their surface. Recall that glyco means sugar, so these are proteins with sugars attached. Red blood cells have glycoprotein A or glycoprotein B. It follows then that type A red blood cells have A glycoproteins, while type B red blood cells have B glycoproteins. Type O red blood cells have neither A nor B glycoproteins. Type AB red blood cells have one A and one B allele, and this leads to both A and B glycoproteins being on the surface of the red blood cells. So type AB blood is an example of codominance. The rest of the blood types are not codominant. Being homozygous or heterozygous for A or B blood yields the A or B blood type with the A or B glycoprotein. A and B are dominant to the O allele, which is why heterozygous individuals that have one O allele have either type A or type B blood. Having one A allele and one B allele together yields the codominant AB blood type with both glycoproteins. Being homozygous for O yields O blood type with neither glycoprotein. Another difference to Mendel's rules is pleiotropy. In pleiotropy, one gene has multiple effects on the phenotype of an organism. An example is Marfan syndrome, which has many symptoms, all caused by a mutant connective tissue protein. Connective tissues run through the entire body, so one gene that changes this one tissue leads to many symptoms. In pea plants, one gene would only have an effect on one phenotype. We now turn our attention to the sex chromosomes. Recall that females have two X chromosomes while males have an X and a Y. Our first question is, how is the sex of offspring determined? If a sperm carrying an X chromosome fertilizes an egg, the offspring will be female. Keep in mind that eggs always carry an X chromosome because eggs come from females and females only have X chromosomes. If a sperm carrying a Y chromosome fertilizes an egg, the offspring will be male. What this means is that sex determination in the offspring is determined 100% by males. Females only have X chromosomes, so will pass down an X to all offspring. Males have a 50-50 chance of putting an X or Y in a sperm cell. If a sperm with an X fertilizes an egg, the child will be female. If a sperm with a Y fertilizes an egg, the child will be male. Therefore, sex determination depends solely on the father. Every time fertilization takes place, there is the same 50-50 chance of conceiving a son or a daughter. Sex-linked genes refers to genes found on the sex chromosomes. Genes on the Y chromosome, which are Y-linked, are only found in males because, of course, only males have Y chromosomes. Genes on the X chromosome, which are X-linked, are found in both males and females. Females have two copies and therefore two alleles of X-linked genes, while males only have one copy and therefore one allele of X-linked genes. X-linked genes in females follow the normal dominant recessive patterns that we've been discussing, since there's a homologous pair of X chromosomes with pairs of genes. X-linked genes in males are visible as traits regardless of whether the allele is dominant or recessive. This is because males only have one X chromosome. Therefore, X-linked recessive traits are more prevalent in males because females need two recessive alleles to have the trait, while males only need one. Example of X-linked recessive traits that are more common in males include red-green color blindness, in which an individual cannot see red or green colors, and hemophilia, which is a blood clotting disorder, in which an individual can continue bleeding after a small cut. The son of a woman who is carrying a recessive allele for an X-linked disorder will have a 50% chance of being affected. A daughter will not be affected, but she will have a 50% chance of being a carrier like her mother. Individuals with red-green color blindness will not be able to see the red 29 on the green background, as all of the dots will appear gray. Geneticists study the inheritance pattern of normal and disease-causing alleles in humans by searching medical, historical, and family records. Traits can be studied over many generations for a family using a pedigree. This is a diagram that shows genetic relationships in a family. Genes on autosomes can be inherited in an autosomal dominant or autosomal recessive pattern. This is a pedigree for alkaptonuria, which is a recessive metabolic disorder. In a pedigree, males are indicated by squares and females by circles. Individuals who are affected with the disorder are indicated by a darker color, while unaffected individuals are indicated by a lighter color. In this case, individuals who are homozygous recessive have the disorder, while homozygous dominant and heterozygous individuals are unaffected. A question mark indicates that the second allele of a genotype cannot be determined from this pedigree. The child in this photo has albinism, in which melanin pigment is not produced in the skin, hair, or eyes. This is another example of an autosomal recessive trait. Polygenic refers to a phenotype that's influenced by many genes. Poly means many, and genic refers to genes. Once again, in Mendel's pea plants, only one gene affected one phenotype. Examples of polygenic genes in humans include height, weight, eye color, and skin color. You have many different variations of all of these traits, and this indicates that many genes essentially have a blending effect to come together to give the final phenotype in an individual. Skin color is determined by the additive effects of many pigment genes that blend to yield the final shade of skin color. Here we examine the effect of the environment on traits. Mendel's pea plants were not affected by the environment. It didn't matter what the environmental conditions were. Pea seeds were either green or yellow, round or wrinkled, etc. There are many traits in many organisms, however, that are affected by the environment. The environment can certainly have an effect on traits in some organisms. Examples in humans include sun exposure making skin darker and hair dye altering hair color. If you lay out in the sun in the middle of the summer, your skin cells respond by producing more melanin, which is a pigment that filters out ultraviolet radiation and will make your skin darker, and you can dye your hair any color you want to. An example in Siamese cats is an enzyme that's required for pigment production is only active in cooler body parts. A Siamese cat has pigmentation in their cooler extremities, the ears, nose, paws, and tail, because the enzyme required for pigment production is only active in a cooler environment. The core body temperature is too warm for this enzyme to be active.